Hello, everyone, and welcome to the Lab Investigations Lightning Talk session. Today, we'll hear from 10 great speakers. Each speaker will present for 3 minutes, and then we'll have an additional 2 minutes to answer any questions. I won't take more time, so first we'll hear from Ashleigh van Heerden from the University of Pretoria. The floor is yours, Ashleigh.
Hello, everyone. So most antimalarial compounds are identified through phenotypic screening, and because of this, the mode of action of these compounds remain largely unknown. This leads to a lot of investment resulting in such as time and costs into compounds that might be discontinued due to them later on showing very undesirable mode of action later on. To combat this, our group wanted to create a platform for early mode of action investigation of hit and lead compounds to accelerate antimalarial drug discovery. For this platform, we wanted to use transcriptomic data, as we saw that within the gene expression profiles of asexual malaria parasites, that different drug treatments showed similar perturbed expression patterns due to different compounds having a similar mode of action. However, because transcriptomic data is so complex and big, we wanted to utilize machine learning, as machine learning has been shown to have to be very efficient in analyzing these big and complex data sets to identify patterns and make accurate models from. So the general idea was to make these machine learning models that would analyze the transcriptomic data of drug-treated parasites and use their transcriptomic data to then analyze what mode of action those drugs belong to. Now, from the models that we built, multinomial logistic regression was the most accurate and stable model. So we utilized this model to rank the genes then and see which genes contributed the most in helping the model identify the mode of action. And from this, we identified 50 biomarker genes that could function as mode of action fingerprints. Now, in the right top corner, you can see when we visualized these fingerprints, we saw similar fingerprints for compounds that had similar drug targets and mode of action. And this is largely when we look at the perturbed expression of these 50 biomarker genes at the bottom left. We see that there is a strong correlation between perturbed expression within these 50 biomarkers between compounds with similar mode of action and an anti-correlation with those with dissimilar mode of action. Interestingly enough, these 50 biomarker genes were identified from asexual data, but when we looked at the expression of these genes in immature gametocytes, we saw that for compounds that were treated in asexuals, we kind of get the similar fingerprint within immature gametocytes treated with the same compound. Therefore, this study contributes a new sensitive strategy to specifically and rapidly identify the mode of action of novel compounds and holds promise to accelerate antimalarial drug discovery programs. Thank you.
All right, thank you. We have a couple of minutes for questions. Please raise your hand if you would like to ask a question or write it in the chat box. Okay, thank you, Ashleigh.
Thank you.
If there are no questions, we can move on to our next speaker, Deepti Shrivastava from CSRI CDRI. Feel free to start whenever you're ready, Deepti.
So good afternoon to all. I'll be discussing about one of the ABC transporter of Plasmodium falciparum, that is MDR6, and it is unique to apicomplexes. And it is predicted to be essential in blood stage. Further, it is a putative iron sulfur cluster transporter as seen in its yeast and human homologue. So towards this, we performed immunofluorescence assay, and the MDR6 was localized to parasite mitochondrion, and it was also confirmed through organelle fractionation assay. Also, MDR6 is an ABCB superfamily member, and in vivo they worked as half transporter, and in an active form they form full transporter, that is a dimeric form. So in vitro we saw on native page it was purified as a homodimer that was confirmed through transmission electron microscopy. Further, through homology modeling and using Swiss modeler, we saw it was a model came of a dimer, and the model was made on yeast template. Then further, we have seen that MDR6 ATPase is an ABC transporter, and in context of ATPase activity, its ATPase activity was stimulated in the presence of oxidized glutathione as compared to reduced glutathione. Further, in the context of iron sulfur cluster transporter, we performed UV visible scan, and we see a clear peak of at 420 nanometer, which is indicative of 4Fe4S cluster peak. Further, we have seen that in the presence of FES cluster and the ligand, the ATPase activity of the transporter stimulated, suggesting that there must be some conformational role played by the iron sulfur cluster in the activity of MDR6. Further, to we dig out some interaction partners of MDR6, and after pull down, it was observed that MDR6 interacts with components of ISC pathway of the mitochondrion. Then we checked that whether the carrier protein of ISC pathway, that is ISCU, is able to transfer 4Fe4S cluster to the upper form of MDR6, and some amount of transfer occurred. So interestingly, we are now on this question whether this MDR6 is the transporter that links mitochondria to the cytosol for iron sulfur biogenesis. Then further, we came across a paper that says there is a significant association between the number of asparagine residues and its drug susceptibility. So for that, we gave lumefantrine treatment to the sensitive and the resistant line, and through Western blotting, we observed that there is a decrease in the expression of MDR6 in a sensitive line as compared to resistant line. And further, through quantitative PCR, we saw that there was no change in the transcript levels, suggesting that lumefantrine is inhibiting the protein synthesis, of playing some role in the expression level of MDR6 at translation level. So suggesting these 2 points, we are on the question that whether MDR6 could be used as a putative drug target, and also is it a link between mitochondria or cytosol for iron sulfur biogenesis? Thank you.
Thank you. We have 1 minute in case anyone would like to ask a question. Ashleigh is asking, is this a membrane protein?
Yeah, it's a membrane protein. So it is an integral membrane protein consisting of 6 transmembrane domains in each monomer.
Okay, thank you. If there are no more questions, we'll move on to our next speaker, Kieran Tebben from the University of Maryland. The floor is yours.
Thanks. I'm Kieran Tebben. I'm a second-year grad student in the Institute for Genome Sciences at the University of Maryland. I'm going to tell you about how we use gene expression deconvolution to study parasite gene expression directly from patient samples. So one of the main challenges in doing gene expression studies directly from patient samples is over here on the left. You can see that each blood sample has a unique mixture of developmental stages inside of it. So when we do bulk RNA-seq analyses on this, some of our results are confounded by the fact that there's different populations of cells that make up each sample. So what we've done is we've used a PPRGI single-cell RNA-seq data set to make a species-agnostic reference data set that we can use to estimate the relative proportion of each cell type in each sample. So I've been using a software called CyberSort-X, which takes this bulk mixture, which you can see on the left, and uses a algorithm called gene expression deconvolution to estimate the relative proportion of each developmental stage in each blood sample. And we can use these proportions to correct our gene expression analyses for the relative proportion of cell types in each sample. And if you look way over here on the right, you can see a graph of the resolution of these results. And we used flow sorted parasites at different time points. And you can see that this does recapitulate the developmental cycle of the parasites pretty well, where you see over here at the 4-hour time points, we see mostly rings, then we move into mostly trophs, and then we move into mostly schizonts. So we can pretty reliably deconvolute parasites into the stages that we expect to be present. So we expect our results to be accurate for patient samples as well. And then down here in this gray box, we've applied this strategy to some samples. 3 Malian children consecutively infected with P. falciparum and P. ovale infections. And you can see that we have run this deconvolution algorithm, and there are some differences in the presence or in the cell types present in the samples. And then we've used these relative proportions to correct our gene expression analyses to try and compare Plasmodium gene expression during infections with different species. So feel free to contact me with any questions. We've also applied this strategy to human cell types. So I also have a lot of data on the human immune response and human cell types that make up each sample. And I'm now applying this strategy to a much larger sample size as well.
Thank you. Sorry for muting you, by the way. That was me.
That's okay.
So we have a couple of minutes for questions. Purnima, if you would like to unmute yourself to ask your question.
Yes, hi. Nice talk, Kieran. So my question is there species differences in state-specific transcriptional analysis that you've done?
So that's sort of what we were trying to answer with this study down here in the gray box, is whether or not there are species-specific differences. We know that we see species-specific differences in the stages present in the blood, particularly in falciparum, we mostly see rings, and in non-falciparum infections, we see a really heterogeneous mixture of stages. But we are not especially sure of specific gene expression differences between species, and that's sort of the rationale for doing this study in the first place.
Thank you.
We still have 1 minute in case anyone would like to ask an additional question.
I have a question.
Okay, sorry about that. I think that Jürgen Bosch raised his hand first.
Okay.
Jürgen?
I was just curious, how much sample do you need from a patient to be able to run a useful analysis?
So when I did my study, I used 1.5 mils of blood in PaxGene. I think the total sample was about 4 mils of blood in PaxGene solution. So I used about a quarter of it for my analyses. So surprisingly little material.
So about 1 mil of blood is what you effectively need.
Yeah, roughly.
Okay.
We could get away
Thanks.
with less, but that's what we use to optimize our RNA-seq.
Okay.
Slavika, sorry about that. If you would like to write your question in the chat box and Kieran can answer you in the chat box, please. We have to move on to our next speaker.
Okay.
Thank you. Great. Our next speaker is Kutub Ashraf from Sorbonne University. Whenever you're ready.
Okay, good afternoon to all. Today I'm going to describe about my research work where we prepared infusions from 2 artemisinin plants and we tested in the Plasmodium liver stages. So till that, artemisinin combination therapies, ICTs, are the frontline treatment against the malaria worldwide. But this life-saving drug is obtained from the artemisinin plants. But it has some, ironically, these ACTs are declining their efficacy not only in the Southeast Asia, but now recent emergence in a part of Africa. And also, these ACTs are not effective against the hypnozoite, which cause the Plasmodium relapsing malaria, especially in the cases of vivaxes, and which compromise the vivax eradication effort. Also, primaquine and tafenoquine are the only licensed anti-relapsing drugs that has been used more than 70 years, but this causes fatal hemolysis among the G6PD deficient patients. And also different mathematical modeling and also genetic analysis of the vivax infections suggests that more than 80 to 90% infections are due to the hypnozoite relapses. So we desperately need novel drugs that can cure relapsing malaria. And to combat this kind of future uncertainties, natural products can be a good avenue for the drug developments, and also alternatively target-based approaches are gaining momentum nowadays. So in our liver stage assay, first we optimize the infusions prepared from Artemisia plants. One contain artemisinin, one is Artemisinin annua, which contains significant amount of artemisinin, and one doesn't contain artemisinin, which is Afra. And then we prepared infusions, and then we tested these infusions in the Plasmodium liver stage, utilizing two cellular models, primary hepatocytes, human hepatocytes, and primary semen hepatocytes. And we incubated this infusion exposed to the infected culture for 3 to 5 days with the strategies of the prophylactic SI and also for the reticle cure SI. And finally, we analyze our data in the high content imaging platform. As you can see this in the findings, this is the findings of the inhibitory activity of the artemisinin infusions that was tested in the Plasmodium liver stages. As you can see that the Plasmodium falciparum is not clear, although, but I can tell you that with the infusions prepared with the 10 gram per liter of NUI and Afra 4 gram per liter, this is the 100% inhibition of the Plasmodium falciparum shygens. And when we expanded our observations into the relapsing malaria, which is Plasmodium vivax, in the human cellular models, you can see that both shygens, the shygens is black and the gray is hypnozoites. Almost 70% shygens and hypnozoite deployment were blocked with the infusion treatments. And also when we, this observation was also accelerated in the cynomolgia models, utilizing the semen hepatocytes. You can see that both shygens and hypnozoite deployment were blocked by the infusion treatment, both the N1 Afra. And here I can say that we also used DHA, which is a prodrug of the artemisinin derivatives. And you can see that with DHA, this in the left one, shows the minimal activity, but other liver stage antimalarial drug like atovaquone and primaquine has significant inhibitions like the infusions. And also in the left panel, in the right panel, you can see that the remaining parasite that were not cleared by the infusion treatment at low doses also has significant smaller parasite sizes. Then we are wondering what could be the targets of organelles by the infusion treatments. And it's previously known that the artemisinin infusions have some essential activities like herbicide, antibacterial, and anti-apic complex and properties. And also it indicates that there is existence of essential traits here between plants, bacteria, and apicomplex and parasite that could be targeted by the infusions. And also some herbicides also have potent activity over the plasmid disruptions. So we do the extensively studied the intracellular localizations morphology of the apicoplast in the Plasmodium falciparum. You can see that in the findings too. As you can see in the left panel, in the schematic figure, you can see the normal apicoplast, where in the normal apicoplast, there is a huge network of the apicoplast branched and also increasingly branched. And we also found other two types of apicoplast morphologies, which are aggregates, the apicoplast machineries, and also void, which has no apicoplast. And these two types,
I'm sorry to interrupt, but we have to move on to our next speaker. Thank you. To anyone, feel free to use the chat box in case you would like to ask a question. Up next, we have Ritu Gill from Maharshi Dayanand University. Feel free to start whenever you're ready.
Yeah, thank you so much. Namaskar, everyone. Myself, Ritu Gill from Maharshi Dayanand University, Rohtak, India. I feel privileged to be part of this Malaria Day Symposium. I am very much thankful to the organizer for giving me opportunity to present my work on correlation of genomic and molecular analysis of Plasmodium falciparum geotin-related factor 1. ZRF1 is an eukaryote-specific, multi-domain, functionally versatile protein. It is characterized by an interesting domain composition, that is DNA-J, ubiquitin-binding domain, UBD, charged region, followed by ribosome-associated domain, RAC head, and at C-terminus, 2 send domains. Its Plasmodium counterpart is 939 amino acid long protein that is annotated as DNA binding chaperone. ZRF1 is conserved and syntenic throughout all the Plasmodium species. We explored orthologs of ZRF1 across 258 organisms, and ZRF1 was found to be highly conserved except missing in few exquisite groups. The multi-domain composition was observed consistently from lower to higher eukaryotes, although we could observe species-specific domain insertions, deletions, and modification. For example, SAN domain is lost in fungi, oomycetes, ascomycetes, and basidiomycetes. Further, rag head is highly variable in sequence and size. Species-specific insertions and deletions, they are prominent in rag head, for example, Plasmodium species. PFZRF1 is a longer protein as compared to human host and is characterized by parasite-specific insertions. These insertions mainly belong to N-terminus region, DNA G domain, rag head domain, while UBD, Geotin homology domain, and SAN domain, they are comparatively conserved in Plasmodium falciparum. DNA G domain, the defining feature of ZRF1 deciphered approximately 50 amino acid long insertion in loop region, that is between helix 2 and helix 3, just before the HSP70 binding pocket. Further, these insertions in DNA G are genus specific or family specific. And this 50 amino acid long insertion was observed throughout all the Plasmodium species. Plant specific insertions were also observed. Further, we located UBD domain in PFZRF1 in comparison to its human host. We generated UBD domain HMM logo from 282 sequences. The generated logo emphasized conserved patches of aromatic amino acid, especially in the central region, with absolute conservation of arginine at position 70. Different constructs of ZRS1 were cloned, expressed, purified, antibodies were generated, relative expression and localization experiments were performed in Plasmodium falciparum erythrocytic stages, that is ring, trophozoite, and schizones. ZRF1 was found to be upregulated in trophozoite stage. IFA experiments revealed differential localization of ZRF1 shuttling between cytoplasm and nucleus in different asexual stages. In a nutshell, the present study provides a detailed analysis of ZRF1, highlighting the species-specific and parasite-specific features that can be further explored for its potential as an antimalarial drug target. Thank you so much.
Thank you. We have time for a couple of questions. Again, feel free to raise your hand if you have any questions. We'll just give the audience a few more seconds in case they are typing their questions. Okay, if there are no questions, thank you, Ritu. And moving on to our next speaker, we'll now hear from Karli Redinger from Case Western Reserve University. The floor is yours.
All right, thank you. Good afternoon, everyone. My name is Karli Redinger, and I'm a third-year undergraduate at Case Western Reserve University, working in the labs of Drs. Jürgen Bosch and Christopher King. And the projects we've been working on are in regard to identifying and developing antibodies against key invasion receptors for Plasmodium vivax malaria. And specifically, we've developed polyclonal and monoclonal antibodies against well-established invasion receptors, such as the Duffy binding protein, apical membrane, antigen 1, erythrocyte binding protein 2, and others for the eventual development of a rapid diagnosis test, as well as other disease detection and treatment techniques. And this is an area of critical importance, especially for P. vivax malaria, due to the lack of current effective techniques for vivax-specific infection detection and treatment, which requires additional medication to fully eradicate liver stage hypnozoites. And we're currently in the antibody validation stage of the project, which involves a variety of lab techniques to determine that these antibodies do in fact bind to their intended targets, as well as to test specificity and affinity to P. vivax invasion receptors as opposed to other Plasmodium species. So starting with our polyclonal antibodies, our initial findings using image stream analysis, which is shown in panel C, specifically for PVDBP, indicate that the antibodies do in fact recognize and bind to infected P. vivax cells at various stages of the parasite life cycle, while showing low binding affinity for uninfected cells. And additionally, paper-based and ELISA assays using mixed P. vivax and P. falciparum cultures suggests that these antibodies have a higher affinity for P5X antigens as opposed to P-falciparum. And based on these assays, our proposed limit of detection for P5X is significantly lower than that of current rapid diagnosis tests that are used in the field today, which is indicated by the graph in panel B. And more recently, with our generated monoclonal antibodies, we've been performing invasion inhibition assays using P-falciparum and P-nolzi with transgenic PV-DBP as models, since we don't currently have P5X culture in the lab. And as you can see in panels A and D, the monoclonal antibodies with greatest blocking ability, which was calculated by % parasite growth after 2 invasion cycles and are depicted either as blue squares in panel A or red squares in panel D, were then selected for dose response assays. And the dose response results from a particular AMA1 monoclonal antibody is shown in panel E, which shows promising results as an invasion inhibitor within transgenic P-nolzi culture. And using these models is also beneficial to analyze cross-species reactivity for certain monoclonal antibodies, which could indicate potential candidates for a cross-species malaria vaccine. And we plan on continuing these invasion assays with monoclonal antibodies within P5X culture to determine their sensitivity for the species in which they were selected. And results from these collective experiments will then be analyzed to determine which of our polyclonal and monoclonal antibodies have the highest specificity for P5X for use in eventual diagnostics and disease treatment. And more generally, just to bring new insight into P5X invasion into red blood cells, which there is currently limited information. So please feel free to reach out to me or my PI Jürgen Bosch if you have any specific questions or would like to learn more information about our project, but I'm also happy to answer any questions that you may have if we have any time remaining.
Thank you. Yeah, we have a couple of minutes for questions. Again, you can raise your hand or type it in the chat box. Okay, if there are no questions, thank you, Karli. Moving on to our next speaker is David Otieno from KEMRI, Center for Global Health Research. Whenever you're ready, David.
Okay, thank you. I'm David Otieno from KEMRI, Kisumu in Kenya, and my presentation is about field evaluation of breast biomarkers for malaria diagnosis. And historically, malaria diagnosis requires microscopic evaluation of patient blood samples, which is slow and requires sterile supplies, equipment, and trained personnel. Quantitative PCR and other nucleic acid-based tests are highly sensitive and specific, but are costly, cumbersome, and require special training and equipment. Rapid diagnostics tests are increasingly available to detect blood malaria antigens and are based on the detection of histidine-rich protein 2. And unfortunately, the HRP2-based RDTs possess critical weakness. Blood sampling for RDTs demands sterile supplies and therefore inadequate for population or border screening issues. In clinical use, false positive RDTs are common because HRP2 is detectable up to 1 month after malaria infection and treatment, such that healthcare providers cannot reliably distinguish acute from recent malaria. Even more troubling, false negative RDTs are also increasingly as rising use of RDTs has selected for P. falciparum strains that lack HRP2 in India, Peru, and Africa, leading to direct call by WHO in 2016 to identify new targets, new test antigens. Finally, as resistance continues to spread to artemisinin-based antimalarials, clinical tests that respond to changes in parasitemia will be critical to evaluating therapeutic response, as low parasite clearance rates are the hallmark for antimalarial resistance. In our published studies, we determined that malaria-infected red blood cell release parasite-specific volatile organic compounds, including terpenes. One malaria-specific volatile organic compound was confirmed as monoterpene alpha-pinene. By comparison to a purified standard, pinene production was reduced in parasites treated with an inhibitor of parasite isoprenoid biosynthesis. confirming that parasite metabolism is required in terpene production. Interestingly, recent work by Amy has established that these parasite-induced volatiles, including alpha-pinene, enhance attraction to mosquitoes that transmit malaria. We therefore hypothesize that volatile organic compounds produced during natural malaria infection will distinguish infected from uninfected children. This hypothesis is additionally supported by preliminary studies demonstrating volatile organic compounds specific to malaria-infected mice and experimentally infected adults. We therefore collected and analyzed blood samples from cohort pediatric patients with and without uncomplicated P. falciparum malaria. In brief, improving pregnancy outcome with independent preventive treatment in Africa children aged 4 to 16 who presented for care at Kamuzu Central Hospital and Boyle Health Center in Lilongwe, Malawi, were consented and enrolled. We found significant increases in monoterpenes, including alpha-pinene, were present in children with malaria. Monoterpene abundance was highly specific, up to 94% specificity for malaria infection. These findings strongly suggest that volatile compounds observed in vitro are likely to have in vivo breath correlates. In an unbiased approach, we also found that as few as 6 breath compounds are sufficient to correctly diagnose malaria with 82.9% accuracy. Not only do these compounds represent potential biomarkers, but these analyses are also highlight that there are global changes to the malaria-specific breath print, which may be harnessed by electronic nose technology. Our short-term objectives are to determine the reproducible changes in breath composition in response to malaria infection, parasite loads, and parasite developmental stages. These investigations will be significant because we will determine the reproducible changes in breath composition in response to malaria infection, which will greatly advance our fundamental understanding of malaria-specific volatile organic compounds. We will also determine whether the breath volatile organic compounds correlate with parasite numbers or life cycle. This information will have high utility in malaria elimination efforts to reduce human reservoirs of malaria transmission through test and treat public health intervention. Malaria-specific volatile will represent novel candidate biomarkers of malaria infection. This basis for future non-invasive diagnostics. Thank you, and if you have any question, you can ask.
Thank you, David. Moving on, our next speaker is Victor Cardoso-Jaime from Johns Hopkins Bloomberg School of Public Health. The floor is yours, Victor.
Thank you. Hemocytes are the most important cells for the immune system in insects, and in the mosquito they produce immune factors that affect the plasmodium development, like TIP, PPO, antimicrobial peptides, etc. However, additionally, they can produce agonist factors such as lectins. Here we evaluate the role of hemocyte during Plasmodium falciparum infection in the mosquito Anopheles gambiae. To do it, we depleted the hemocyte through previously reported methodology that consists in inject into the abdomen of the mosquitoes liposomes that contains cloldronate. And you can see here that this methodology deplete around 90% of the hemocyte. Then we evaluate the effect of the hemocyte depletion in the infection of the Plasmodium falciparum in Anopheles gambiae. And you can see here that in median infection, we didn't observe any change in the number of oocysts in the midgut. However, when we supplement the sucrose diet with antibiotics, we observe a decrease in the mosquitoes with hemocyte depleted. And that means that probably hemocytes are protected to the parasite in a microbiota-dependent way. In addition, we want to test if the intensity of the infection is affected by the hemocyte depletion. And we observe that the high doses of the Plasmodium falciparum, we observe a decreasing of the number of the oocysts in the midgut in mosquitoes with hemocyte depletion, suggesting that the hemocyte are protecting to the parasite at high doses levels. Surprisingly, we observe a high mortality in mosquitoes with hemocyte depletion after Plasmodium falciparum infection, but this mortality it's decreased when mosquitoes are treated with antibiotics, suggesting that the mortality probably is associated with over microbial load. Surprisingly, we analyzed the bacterial load in the midwood in the mosquitoes with Hemocyte depleted, and we observed that in these mosquitoes, an increase in the bacterial load in the midwood, suggesting that the Hemocytes are regulating the bacterial load in the midwood in some way. Also, we observed that blood feedings by itself can induce high mortality in the mosquitoes with the Hemocyte depletion, suggesting that the hemocytes are really important for the mosquitoes against bacteria, but also this effect or this this effect is affecting the bacteria and the parasite in opposite way. And that's it. You have some question?
Thank you, Victor. Do we have any questions? Once again, raise your hand or write in the chat box. Yep.
One quick question: what's the percentage of antibiotics, and what type of antibiotics have you used for this?
Oh yeah, penicillin and streptomycin for cell culture. Yeah.
Okay, thank you.
You're welcome. Thank you.
Any additional questions? We'll just give him a few more seconds. All right. If there are no more questions, thank you, Victor.
Thank you.
Yep. Up next, we'll hear from Sharon Patray, also from the Johns Hopkins Bloomberg School of Public Health. Feel free to start whenever you're ready, Sharon.
Hi, I'm Sharon. I'm a PhD student from the CNS lab, and I'll be presenting my work on the investigation I did into CSP trafficking. CSP is the circumspozoid protein, also known as CSP, can be seen coating the surface of the sporozoa stage of the parasite very densely. If I can draw your attention to the image on the far left under the hypothesis, CSP is stained in red, and you see it's densely coating the parasite surface, but there isn't a lot known about this protein and how it is trafficked. CSP can also be seen shed in the parasite trails as the parasite moved. And one of the things we're interested in this was if CSP goes through the Golgi as it's trafficked to the surface of the parasite where it's shed. And we hypothesized that because due to the small size of the Golgi in the stage of the parasite, and the Golgi is represented in that very same image at the bottom in green. You see the two little dots on either side of the nucleus, and that is the Golgi in this stage of the parasite. And because of this small size of the Golgi, that this protein is such abundantly seen on the surface and shed, probably doesn't go through the Golgi. To study this, we use the compound bifardin A, which inhibits trafficking from ER to Golgi, and in the process blocking or most trafficking to the plasma membrane. Our plan was to use bifardin A to observe the effect on CSP shedding. To do this, we first did a live death assay to assess the effect of bifardin A on the parasite. And as you can see from the image in the middle on the top, we have the heat-killed parasites which are stained with CSP in red and the live dead dye in green. And they are represented in orange, green and orange color. And then the minus bifardin CSP, and then the bifardin treated at 0 hour and then 3 hours are all in red, which shows that they are alive. And then quantification below that also show the percent dead. So after we were confident that the Felding A wasn't killing these parasites after 3 hours of treatment, we then did a pulse to look at newly synthesized CSP over a 3-hour period in the presence and absence of the Felding A. After that pulse, we separated the shed content that would be in the supernatant from the parasite pellet. The parasite pellet was then washed and lysed, and both the shed supernatant and the parasite lysate were IP'd for CSP and TRAP. If I draw your attention to the image at the top of the right-hand panel, you will see that TRAP is also a protein that is shed in this stage of the parasite, and a little more is known about TRAP trafficking. Like it's known that it goes through the microname, and therefore we could also predict that if it goes to the microname, it most likely goes through the Golgi. So we use TRAP as our positive control in this experiment. The graphs at the right beneath those images are the audio radiograph from the illusion of these experiments. On the left-hand side, we have the TRAP graph, plus and minus Felding A. Minus Felding A is on the left-hand side. In that experiment, I loaded the whole lysate, the flow through after the IP, and then you have PE, which is representing the parasite lysate illusion from the IP, and then you have SE representing the shed supernatant illusion. And as you can see, we see a band in the minus Felding for TRAP. We see a band with the parasite representing the whole TRAP. And when TRAP shedding occurs, it's a cleavage event. The protein is cleaved from the C-terminus end away from the parasite, and that is represented in a smaller molecular weight band. I've labeled SE, and when we add the Felding A, you see that SE band disappears, and we see a lot of TRAP in the PE band that's not being able to be trafficked out of the parasite.
I'm sorry to interrupt, Sharon, but we have to move on to our last speaker.
Okay.
Thank you. Moving on to our last speaker, we'll now hear from TM Andrew Dinchi from the National Veterinary Research Institute in Nigeria. I hope I didn't butcher your name, by the way, but okay, the floor is yours.
Good evening, good morning. I am Dinchi Andrew TM from the National Veterinary Research Institute. I'm going to present a paper on the nested PCR analysis of Plasmodium parasite in malaria infection in northern Nigeria of Nigeria. Malaria is placed among the top diseases of serious concern in Nigeria. As everybody knows, all the human malaria parasites are prevalent in the northern region of Nigeria. But our concern is that the current laboratory diagnostic technique, which are RDT and microscopy, do not detect all these Plasmodium species due to some factors, human factors, and some errors. The main aim of this study was to validate a nested PCR for the detection of human Plasmodium species. And the second aim is to study the occurrence of these non-Plasmodium species in the northern region of Nigeria and to compare the results obtained from the classical laboratory results obtained in the region, and then to now validate the nested PCR for detecting human Plasmodium species in the region. A total number of 302 positive malaria blood samples were collected from 11 healthcare centers in 4 states: Plateau State, Nasarawa State, Kaduna State, and then the FCT. And we collected them based on the current laboratory technique they were using, which were microscopy and RDTs from those regions. For our work, we used 13 samples selected to validate the PCR protocol that we were adopting. And 2 out of these 13 samples gave us a distinctive and specific 205 base pair band, indicative that those samples were Plasmodium falciparum, which we use as our positive control. We went further also to run specificity and specificity tests on some of the samples we had. In this study currently, out of the 302 positive samples we collected, 291 were based on microscopy results, and only 11 were RDT positive. And then we analyzed these samples by the nested PCR. And out of these 302 positive samples that we collected based on microscopy and RDT, only 76 samples were positive for falciparum, and only 3 were positive for Plasmodium ovale. We did not get vivax, we didn't get malaria from these samples we analyzed. So for the specificity test, which we did a 10-fold serial dilution, and we got at tube 5, which is 10 raised to the power of 5, gave us a limit detection of 0.99 microgram concentration of DNA. So at that point, we knew that at this concentration, you could detect the Plasmodium parasite using our PCR method, which we validated. Then we went ahead to run specificity tests. We used DNAs of Trypanosome brucei and Babesia to run alongside with our positive falciparum DNA. And from the result we got, there was no amplification observed in the other hemoparasite used. That means that the primers we used for this work were specific only for P. falciparum. So we went further.
I'm sorry to interrupt. Thank you so much. But we kind of have to stick to the schedule. I would like to thank all of our speakers and everyone else for joining us in this lightning talk session. That's it from me, and I hope to see you all in our next World Malaria Day. Thank you.